Welcome back, everyone. We are discussing our genetic controls that we have in our cells, how we control which genes are turned on and which are turned off. Right now, we're in the middle of discussing some prokaryotic ways that they control their genes. We've discussed the lac operon and the tryptophan operon. And now we're going to talk about the CRISPR-Cas9 system. You may have heard of CRISPR and Cas in terms of things like gene editing, ways that we can study different genetic sequences. But these are stretches of DNA that are found naturally in bacteria and archaea. These little repeat stretches of DNA. They're called CRISPR. That is an acronym for clustered, regularly interspaced, short, palindromic repeats. So we see these little stretches of genes in their DNA that actually originally came from viruses. So these viruses that in that cell's evolutionary history, their ancestors had been exposed to these different viruses. And it's a way that their genome, kind of like almost an immune system, remembers what kinds of viruses it's been exposed to before. Their CRISPR sequences are associated with Cas proteins, or CRISPR-associated proteins. Some of these CRISPR-associated proteins help incorporate viral sequences, so when the cell is exposed to a new virus, it'll be able to build a memory of that virus. Some of these Cas proteins help process their target RNAs and get them into the CRISPR locus. So these CRISPR genes will encode what's called pre-CRISPR RNA or pre-CRRNA, which is going to be made as this nice long stretch of all of these viral RNAs that they're remembering. And then these CRRNAs can be cleaved by Cas proteins, so that they're now into individual little stretches of RNA for each of the different. So like this is the green one for the green gene. You'd have a red one for the red gene for each of those different RNA sequences of the different viruses. So then if a virus from their memory infects them again, that target DNA will be recognized by that CRISPR RNA. The CRISPR RNA will be able to base pair and recognize it. And the Cas sequence is able to then cleave that invading RNA, that viral RNA, so that the cell is not going to be, killed by the viral infection. This ability to cleave a target stretch of a gene is why this works so well for gene editing, because we're able to specifically cleave a gene that we may want to remove or to turn off or to change. All right, so that's a pretty quick explanation of the CRISPR system. I'll see you back for our next video. We're going to start talking about some eukaryotic controls, why it is that different cells of your body behave differently and produce different proteins. I'll see you later. Bye.